So welcome everyone to SADS Living with SADS webinar, where we're going to hear tonight an overview about clinical trials. I'm Marcia Baker, the program director for the SADS Foundation, and we're honored tonight to have Dr. Dominic Abrams of Boston Children's Hospital, who is volunteering his time for this presentation. So it's now my pleasure to introduce him. Dr. Abrams is an adult and pediatric electrophysiologist and has extensive experience in the management of inherited arrhythmias with the entire family. Dr. Abrams is co-director for the Center for Cardiovascular Genetics at Boston Children's Hospital and associate professor at Harvard Medical School. He completed his medical school and postgraduate training in London, England, United Kingdom. Before being recruited to Boston, he held dual hospital appointments in the UK that allowed for detailed evaluation of all family members with varying inherited cardiac conditions, including ion channel disorders and cardiomyopathies. So thank you, Dr. Abrams, for your time tonight to talk about clinical trials. After his presentation, we'll have time for him to address your questions. With so many of you on this webinar, please type your question into the chat box for Dr. Abrams, and we'll address those following the presentation. So I'll turn it over to you, Dr. Abrams.
Marcia, thank you very much for that very generous and kind introduction. And it's a real pleasure to be here. Good evening, everyone, or good afternoon, depending on where you are across the country. And I'm just going to quickly log into my slides. So as you all know, the topic of tonight's webinar is going to be clinical trials, obviously related very much to inherited cardiac conditions. And I'm going to spend about 30 minutes discussing different aspects of this. Really, I hope taking it right back to the beginning so people get an understanding and an overview of how we get to clinical trials, how long the process takes, what the clinical trials mean, and then talking a little bit about sort of how you find out about trials, which one is right for you, etc., etc. And then I'm going to talk, I hope, for about half of the session, and then we'll leave the other half for questions, which is usually obviously an area of much interest, and people can questions about what sort of areas of interest they particularly have. So just to discuss my conflicts, I'm on the scientific advisory boards of two companies, both Rocket Pharma and Thrive Therapeutics, both of whom are involved in this space. So very important that you know that. But also very important, I'm not going to discuss specific trials tonight. I'm going to give a broad overview, but stay away from any specific individual trials, obviously to prevent any further potential conflicts. So I think if you were to sort of Google what does a clinical trial pathway look like, you come up with something a little bit like this, which is a 5-stage process of preclinical research, followed by phase 1, phase 2, and phase 3 trials, and then once the drug is approved, thinking about getting into sort of the post-approval analysis, which continues as the drug is released into the market. And we're going to talk about all these different areas today, but I want to sort of frame it slightly differently and think about, well, how do we get to the position of this preclinical insights of research? What is it that drives thinking about creating a new molecule or a new understanding or a new way to treat a specific disorder? And of course, that all comes from studying patients in the clinic, understanding broader families, and really understanding both the genetic and the molecular basis of different conditions to really drive an understanding of how we can potentially treat them in different ways. So I'm going to focus mainly or think mainly about pharmacological trials tonight. So different pharmacological agents, those could be small molecule therapies, those could be gene therapies, of course, which are being increasingly talked about. But there's many other non-drug trials that have been performed in the past, and I'm sure will be performed in the future. Some of you may be very aware of these or participated in either the LIV-HCM or LIV-LONG-QT syndrome. Those are about lifestyle and exercise in both hypertrophic cardiomyopathy and long QT syndrome, respectively. Lots of studies are being performed in other forms of cardiac conditions, including modifiable risk factors, non-cardiac medications and how those may influence different conditions, diet and nutrition, the microbiome increasingly important, And then, of course, we're now in 2024. So AI and data analytics is going to be, I'm sure, something that rears its head more and more as trials develop. So there's lots of non-pharmacological trials and interventions that are being performed, as well as sort of more standard clinical trials, which involve a specific agent that's been developed. So just to raise that as an important component of things, but not something I'm going to discuss in any great detail further tonight, but obviously very happy to do so in the questions. So I'm always fascinated by the history of medicine and sort of taking it back right to the very beginning. This is a slide related to hypertrophic cardiomyopathy. This was first described by a gentleman called Donald Tier, who was a pathologist working at St. George's Hospital in London. And he reported in 1957 these 9 patients who all had a very similar picture of significant hypertrophy of the ventricle associated with changes on histology and changes on the ECG. So he first reported this, called it asymmetric hypertrophy. And interestingly, in one of these, there was a sibling who seemed to be affected as well. So sort of alluded to potentially a familial basis and therefore a genetic etiology that was driving this disorder. But it was really this gentleman over here on the right called Peter Paré, who was actually a respiratory physician working in Montreal, who identified this large family. You can see once you get into a wagon wheel pedigree, you know it's a very large family, in a town called Quaticook in southern Quebec. I apologize for the pronunciation if there's anyone from Quebec on the call. But he recognized this so-called Quaticook curse. And of course, this was so important because there were so many affected family members that it allowed delineation of the genetic basis of the condition. And ultimately, this was diagnosed as hypertrophic cardiomyopathy related to the myosin heavy chain, or MYH7, which is one of the commonest 2 genetic causes of the condition today. So really, it's all about understanding families and how families inform you about genetics. And despite all the amazing technology we have, going back to the family and reaffirming how the genetics is behaving and using the family as a tool to really accurately define the genetics remains fundamentally important. But of course, in today, we don't sort of get families like this so frequently, but it's really sort of very, very important to understand how these things sort of were originally devised. And then the same happens in many other conditions. So this is arrhythmogenic cardiomyopathy of different forms, arrhythmogenic right ventricular cardiomyopathy related to plakoglobin, and left dominant arrhythmogenic cardiomyopathy related to DSP on the right-hand side of the screen. And these again were 2 autosomal recessive inheritance families. So one in the Greek island of Naxos, the other in Ecuador. And that allowed for what we call linkage analysis of the family to better understand the genetics. So again, understanding the autosomal recessive, autosomal dominant basis of conditions allowed us to really understand what is the genetic makeup, and from that, what is the molecular phenotype of any one specific condition. And of course, this has been done for many other things. Now, long QT syndrome on the left, starting in the mid-1990s, and then catecholaminergic polymorphic VT in the early 2000s. And again, you'll see the importance, particularly on the right-hand slide, of the families and how understanding the family allowed people to understand the genetics. And of course, it's the genetics that gives us a really good background or really good insights into what is the molecular defect that's associated with the condition that we see in the clinic, on ECGs, on echocardiograms, etc. So with hypertrophic cardiomyopathy, it's really important to understand the phenotype. Over here on the left, you can see echocardiograms of significant ventricular hypertrophy in both the 4-chamber view and in the short axis here, looking through the heart. And this is a condition associated with genetic changes in proteins in the sarcomere. This is the sarcomere here. This is the myosin heavy chain, MYH7, that was first, or encoded by MYH7, that was first described back in that original Quebecois family. And understanding how this behaves and what this actually does and what these genetic changes do has really driven a lot of the understanding, and was the sort of the basis of mavacamten, which is a new specific medical therapy for patients with obstructive hypertrophic cardiomyopathy. So it's really being able to understand the genetics, how that translates into proteins in the heart and how those become dysfunctional, and how that translates into a condition that allows us to generate new therapies for any one disorder. So now, of course, we have a pretty broad understanding of lots of aspects of the cardiomyocyte. We've got different components over here on the left, the desmosome associated with various forms of arrhythmogenic cardiomyopathy, the sarcomere, predominantly hypertrophic and dilated cardiomyopathy, and then channels that conduct ions either in and out of the cell or in and out of cellular components such as the sarcoplasmic reticulum that are associated primarily with the inherited arrhythmia syndromes. So it's really been all that highly valuable work 20, 30 years ago in looking at big families, understanding those basic factors that we talked about earlier, that's got us to the place where we are today and has allowed us to sort of really now be in a position to treat these conditions in a very different way. So one thing that's become increasingly important that I thought it was useful to discuss, because it is being used a lot in preclinical research, is induced pluripotent stem cell modeling. And this is something we do a lot here in Boston, and other centers do as well, and it's become a hugely valuable tool. But I thought as this has kind of really been the building blocks for a lot of the clinical trials that are now evolving, it was useful just to briefly inform people about what this was if they weren't familiar with it already. So we identify a patient who we see in the clinic who has a known genotype and a known phenotype. So we understand the genetic basis and we understand the condition they have. They then give us red blood cells. We used to use skin initially, but now we can just use red blood cells. And then using some phenomenal programming, those red blood cells are then transformed back into pluripotent stem cells. And those stem cells are reprogrammed to then form mature cardiomyocytes. So this is why we call them iPS-derived cardiomyocytes. So these are now cells that function, that beat. You can see them beat them. They can be put into layers. And this, of course, is a perfect genetic model of any one individual with an inherited condition. Prior to this technology, which was really first came out in about 2010, 2011, we were very much using animal models, so zebrafish or mice, which were genetically engineered or given a condition. But of course, that's fine, but it's not a human, it's an animal, and also doesn't have all the other genetic variability that we all carry as people, which varies however any sort of disease-associated variant in a gene may behave. So it's really created this perfect model, if you like, of the patient's heart, and has allowed us to study things and has allowed us to really understand what it is, the molecular problems, and how we can address that from a pharmacological perspective. So something that you may see a lot, particularly if you're researching clinical trials, because a lot of the preclinical research will be performed using this technology, which a lot of labs are now using, not just in cardiology, but in lots of different fields in medicine, and has really transformed the way we can understand conditions and do research. So this is just one example of what we did. This is some work on a condition called catecholaminergic polymorphic VT. Many of you may be familiar with this, many not, but by using these induced pluripotent stem cells, we were able to look at the cardiac ranitidine receptor, which is shown here on the slide. We've got the sarcoplasmic reticular membrane at the bottom, the ion channel in the pore region of the protein, which actually allows the calcium in and out of the SR, and then a large intracytoplasmic section with different zones in it, which I've identified as 1, 2, 3, and 4, R1, R2, R3, and R4, which are the functionally really key aspects of the protein where the vast majority of different variants that can cause the condition have been identified. Over on the right of the screen, you'll see two different little letters P, which identify phosphorylation sites. And that's where other proteins and other enzymes interact with the ranitidine receptor and fundamentally affect how it works. The one at the top is phosphorylase kinase A, and the one at the bottom is calmodulin kinase, or CAMK2. And as you can see, these are at different amino acids, serine 2808 or serine 2814, so just 6 amino acids apart. But by looking at these and by studying these, we were really able to understand what it was that was causing the ranitidine receptor to become dysfunctional, at least in some of the patients we were studying, and how we could potentially influence that. So we found that we could create these heart cells, we could induce arrhythmias, and then we could inhibit either the phosphorylation site at 2808 or the phosphorylation site at 28114, either by blocking it with medications or by genetically editing it out. So when we did that, by inhibiting amino acid 2808, nothing really happened. But when we inhibited amino acid 2814, there was a really significant decrease in the arrhythmias, suggesting that inhibition of calmodulin kinase was really key to the dysfunction of the ranitidine receptor in patients with CPVT. So these cells engineered from patients really allowed us to understand some of the molecular issues that were causing the problem. So from then we could go on and develop a specific calmodulin kinase inhibitor. This is all preclinical research, obviously prior to clinical trials occurring. So what we did was make this specific construct you can see up here at the top. And there was an active agent called autocantide 2-related inhibitory peptide, or AIP, that is a very potent inhibitor of calmodulin kinase. This had been known for some time, but the problem with AIP and inhibiting CAM kinase is that it's expressed in multiple parts of the body, particularly in the brain, where it's involved in lots of things such as memory. So it's not something you can just inhibit with a oral or intravenous drug, because it will have multiple effects where you don't want them to happen. So what we were able to do was create a gene therapy strategy where this would bind only specifically to heart cells, would inhibit the calmodulin kinase. And we did this in sort of different situations. We did this in very young mice, in older mice, and then again in iPS cells derived from patients. So by understanding the genetics, understanding the molecular biology, we can then start to test different treatment strategies in different situations, either animal models or human cardiomyocytes, to assess how we can inhibit these different structures in the heart cells. And then we sort of mimicked what would happen during exercise. So we could stimulate the heart with pacing. We could give epinephrine or catecholamine provocation, both to the mice and to the cells. And what we found was by inhibiting calmodulin kinase, we could really significantly suppress the arrhythmias associated in patients with CPVT. And this is all sort of work led by my colleague Vasilios Bezaridis in Boston, who's been really driving this forward. So this really is kind of taking everything from patients that we'd identified in the clinic, knowing their genetics, knowing the molecular abnormalities, and then creating specific agents that we think can then inhibit the dysfunctional parts of the heart cell and prevent the clinical features that we see associated with conditions. So that's the sort of the clinical insights and the preclinical research that we've talked about in some detail. And at that point, when an agent is thought to be appropriate for trials and appropriate to use in humans, it will not have been used in humans up to this point. There's then a sponsor involved. A sponsor may be a company, usually a biotech company or a pharma company, who are sort of interested in trialing this in different situations. And they will then apply to the FDA for what's called an IND, which is an investigational new drug application. And that then allows them, based on all the preclinical research, the clinical insights, the clinical need, the FDA will then approve that, hopefully, and say, yes, you can start to perform clinical trials. And obviously, they will, as part of this proposal, talk about how they're going to do these clinical trials. And there's basically 3 different phases of clinical trials: phase 1, phase 2, and phase 3, that we'll sort of talk about in a little bit more detail now. So you can think of them really quite simply. Phase 1 is usually quite a small trial. These are numbers that really relate a little bit more to sort of much more common conditions than we're talking about this evening. So in phase 1, it's 20 to maybe 100 patients. And these are control subjects who don't have the condition for which this specific agent has been designed. So this is really looking at safety and pharmacokinetics. So patients or individuals who have volunteered to do this are given the treatment. Different things are recorded, different blood tests are performed. At what time does the drug peak in the blood system? All these different things to really understand, firstly, how this agent behaves in the human system, which obviously has not been tested up until this point. And then secondly, is it safe? Are there any side effects that aren't wanted? And all of these are going to have been looked at in great detail in the preclinical studies, but then they want to confirm that that's the case in humans as well. So phase 1 is not a patient-related study. It's control individuals who've kindly volunteered to do the study. And as I said, focus very much on safety. We then move on to phase 2 studies, which is larger, as you can see. In this example, that's sort of 100 to 500 patients. And what we're doing now is acquiring data on patients who have the condition in question. So, if we're treating a specific type of cardiomyopathy, for example, these will be patients with that cardiomyopathy who meet the specific entry requirements of that study. And the entry requirements will be based on to what degree the sponsor and the scientists involved think that this drug is going to be most effective. So this is a relatively small sample. It's usually done in a controlled way, a few centers. And then if that is successful, the company and the sponsor will then move on to a phase 3 study, which is similar, but much bigger. So we're still talking about patients here who have the condition in question, but a much larger and hopefully more diverse mixture of patients who still fulfill the entry criteria, but may come from lots of different situations. So it's a sort of a bigger study to look, is the effect still the same we saw in a small group of patients? Obviously, there's more statistical power in assessing a larger volume of individuals, and they really want to confirm that this is still working as effectively as we thought it was in a smaller group of patients. So moving from phase 1 to phase 2 to phase 3, obviously these things are very carefully assessed and regulated as the study moves through. Any issues or side effects are then reported back to the FDA during this, and the FDA will reassess a huge amount of data based on any concerns, and then allow the study to proceed. And of course, at the end of each phase, the company will report out data about that specific phase and how effective their treatment was. So you're going to get really good information about these specific agents as it moves through the trial phase process. And then if phase 3 is successful, the company involved at this stage will apply to the FDA for a new drug application or an NDA. And that's the FDA's approval of saying this agent can now be released onto the market. This can now be licensed and given to patients who have the condition who are not in a trial setting. So it's a clinically approved agent. And of course, lots of things will continue to happen at that point. There'll be lots of assessment, other studies, maybe some slightly different situations. So everything will still be quite tightly regulated, but you'll get a lot more information on a much broader group of patients because it's now readily available to everyone who has the condition in the sort of the much wider community, and that obviously can be both national and international. So we've now gone through all the way through the different stages from insights to preclinical research through the 3 phases of a trial, and then on to post-approval marketing and continued analysis. And this is sort of phase 4, as we talked a little bit about, which is just continued assessment. You have a product that's now available, and the companies will continue to assess this and analyze this, and may be involved in lots of different studies, potentially comparing with other treatments and other therapies that have been available for longer. So I think, questions people always often ask us, how do we find out about clinical trials? Well, there's lots of different ways to do this. Firstly, ask your cardiologist. Are there any clinical trials that you think are going to be suitable for me? Question we get asked in clinic every week, and it's really great that people are very keen and enthusiastic to find out what's going on in the field, what's actually happening. And we often talk about sort of where we're at with our preclinical research, as well as potential trials that may be up and coming and may be of interest to our patients. If you see a cardiologist who's not involved in these kind of things so much, and not everyone is, and that's fine, you can always contact a more regional center who may have more opportunity to conduct these trials. And obviously you can do that by arranging appointments or online. So lots of different centers will say, these are the clinical trials we're conducting at the moment. These are things that we're enrolling for. And then you may be interested to find out more from them. So there's lots of information available. I just did some Googling this afternoon as I was preparing the slides and just saw what was out there. And a lot of centers will have sort of information about different trials that they're involved in and they're conducting available, and also contact information as to how you can find out more. Obviously, we're all here because of SADS. So the SADS Foundation is fantastic about informing patients and families about different studies that may be of interest. And increasingly, there's a lot in the press. So you can read both the medical press and the lay press, often telling you about things that have happened at conferences, information that's been released, which may give you some very good information as to what's happening in different areas. So big studies will often get picked up and reported in the general press and not just the medical press. And then, of course, there's all sorts of other organizations such as the American Heart Association, the American College of Cardiology, who have great and very informative websites who can also sort of help provide information around different studies that are either ongoing or about to start recruiting. And then the NIH also have a great website called ClinicalTrials.gov, which is where you can look up different studies that are happening. You can put in certain criteria, so specific diagnoses, specific conditions, and it'll tell you different studies that are enrolling at the current time or those that are being completed as well. So lots of ways to find out about different studies, and hopefully you can get all the information that you need from one of those multiple sources. So I think something patients often ask is the clinical trial right for me? Is this the right study? Should I get involved? What are the things I should be thinking about? Well, I think importantly and fundamentally, do I have the right condition? Is this agent specifically targeted to what I have? And often there's different variations on different conditions. So is this sort of, do I have a form of that condition that this agent has been designed to treat? I think increasingly, particularly obviously with gene therapy, we're going to see new molecules or new therapies that are targeted at a specific genotype. So specific genes will be, openly enrolling, or specific studies will be openly enrolling for patients who have a specific genotype associated with that. So again, really sort of stressing the importance of understanding the genetics in any one individual or the family. Secondly, what is the science behind the new compound or the new agent? You can obviously always look that up, find out what's happened, where's it been published, and sort of what is the safety data that may have emerged from the initial phases of phase 1. And often you'll find this on company websites. They will put this out, maybe that's something that they presented at a meeting, or, and sort of talk about, this is our results from phase 1. We're very pleased. We're going to move on to phase 2 with this specific study. Understanding a little bit about the sponsor, that's either a biotech company or a pharmaceutical company. Again, all these companies have great websites with lots of information and often have lots of great patient advocates who you can speak to and understand more about things to sort of get a better idea of both the nature of the study and specifically what it's aimed at. I think it's always a good question to think, well, how will this new agent help me? Is this something that's going to make me feel better? Is this something that's going to change what medication I need to take currently? Is this something that's going to decrease risk? So these are sort of all the important questions I think it's good to ask, really to sort of understand what are the benefits to me as an individual in terms of engaging and enrolling in this trial. And then it's always good to understand what are the obligations in regards to the trial. How many times do I have to go to the hospital? How far away is the hospital? What happens if I need to stay overnight? All these kind of things. And then what are the investigations that are going to be needed? Because often these things are very highly regulated for good reason. So we really understand that this new agent is doing what it should do and not what we don't want it to do. So there's often lots of investigations that are needed as part of this. So really understanding what is the kind of schedule that I'm signing up for here. If I engage in this trial, what am I going to be needed to do? And then what happens after the trial is concluded? If this seems to be very, very successful, and often, or if the trial's been really successful, it may end early because the results are already conclusive. And so is there what's called an open label extension? An open label extension is where you can continue taking the active medication if you've been enrolled in the study, if it's helping you and it's been shown to be statistically beneficial across a large group of patients in a phase 3 study. So this is just a sort of a collection of things that you might want to be thinking about, or sort of questions you might want to be asking to your cardiologist or your local cardiac team who are involved in your care, and sort of thinking about through which clinical trial is the right one for you. And I think things that are sort of changing significantly, I just thought I'd put this in as a little conclusion. This is a thing that many of you may know about. This is a little girl called Myla who had Batten's disease, who was treated at Boston Children's Hospital by a guy called Tim Yu, and they sort of really created this personalized medicine. So we're now I show this really to sort of emphasize that we're getting into now into these N of 1 studies. So we really are getting into a situation whereby we can create sort of customized therapy for individuals. And it's a kind of an incredibly exciting time. There's lots of activity around this, but this was one of the first times this was performed and achieved. And it's a really incredible story. And Tim Yu, you can look him up on YouTube and hear him speak about it. It's a very, very interesting situation. Unfortunately, mine didn't survive, but it was sort of a really, really important step forward in sort of understanding N of 1 therapies and how those can help patients in different situations. So I think we really are entering that era where this highly personalized treatment is going to be sort of increasingly important. So just as a sort of final addendum to discuss that. I'm going to finish there, and very happy to obviously start answering questions that have come in or may come in, and delighted to sort of discuss anything that we've spoken about here or anything that specific anyone would like to ask.
Thank you very much, Dr. Abrams. And everyone is free to post a question in the chat, and we will run through those. You did such a terrific job.
Everyone's...
Here we go. All right. Melanie asks, can you participate in more than one clinical trial? I've heard if you've done one, it disqualifies you from others.
Great question. Typically, you're right. Typically, you can only be sort of enrolled in one trial at a time because there may be potentially complementary effects of different agents or negative effects on different agents. They may interact with each other in different ways. And obviously, what the investigators are really trying to understand is how one specific agent works. And so by having confounding this with another agent, then that can be problematic. That's running simultaneously, obviously. If you've been involved in something in the past, often people can enroll in new studies in a different situation, but it would usually be sort of determined by the specifics of the entry criteria and exclusion criteria in the second study. So I think if you've already completed one study and that's all finished and done, and then you enroll another one, I can't see any reason why that shouldn't be okay. But again, it's going to be determined by the study entry criteria. But simultaneously, it is always going to be complicated for those reasons.
Okay, thank you. And we have a question from David Wise. Is gene therapy effective for late stage ARVC in heart failure?
Another very good question to which I don't think anyone knows the answer just yet. I think we're all waiting to see what happens. And, with the things like gene therapy, it's all about, when do we intervene? When's the best time to do that? And I think all these things are quite unclear at the moment. So I think we'll have to sort of wait a little bit and see how the initial studies, the sort of phase 1s and phase 2s, when those get rolling, how those, or the phase 2, what that shows us about changes in the right ventricle, changes in the left ventricular function as well, before we can able to sort of to concretely answer that question. So more to come, I'm sure, fairly soon.
All right, I have a question from Lori, saying people might want to understand why genetic trials are different. For example, why phase 1 is a smaller number and treatment of patients with diagnosis not health human subjects. I'm not sure I understand that last part. Trials for genetic conditions, especially with gene therapy, does not always follow the typical drug development of phase 1, then 2, then 3, etc. Comments?
Yeah, I mean, every trial is different. I mean, sometimes if, phase 1 is incredibly successful, studies have gone straight to phase 3. But I do think safety is always paramount. So there's always going to be a phase 1 study to make sure that this is safe, make sure it's not having untoward effects that just couldn't be predicted in, preclinical studies. So I think that's always going to be the sort of the bedrock, making sure this is safe. And obviously there'll be a lot of monitoring investigations that happen, EKGs, echoes, etc., to make sure that the sort of potential effects don't happen. So once that happens, and if there's concern about that, then that's much better to try it in healthy, unaffected individuals or individuals who don't have the condition in question before moving on to those who do. So that's why it's structured in that way.
Okay, thank you. Kristen asks, when you're in a clinical trial, can you ever find out whether the agent had an effect on you, not just the aggregate data?
Again, I think that depends on the degree of unblinding at the end of the trial. So what effects, as you said, what did this do to me, rather than just was this sort of an aggregate benefit or an aggregate no change. Again, it would sort of depend on how much of the information is released and how much is open at the end of the trial. But typically things are unblinded and you can sort of get information related to that. But again, it would all a little bit dependent on the specifics of the study. But ideally, it would be nice to know that. But once it goes into open label, then obviously you can start to get a lot more information about what's happening and how it's benefiting any one individual.
Okay, Andrea asks, can you share any insights about the current PKP2 trials and the impact those may have on DSP and other gene therapies?
So I guess I think thinking about that in two ways, whether gene therapy with trials related to plakophilin 2 will impact patients with genetic changes in DSP or other desmosomal proteins associated with arrhythmogenic cardiomyopathy. We wouldn't assume that they would, but sometimes you can use other proteins to enhance or solidify or to prevent dysfunction of neighboring proteins that they bind to. That's been looked at in lots of different situations before and may be so in clinical trials. So because these are typically haploinsufficiencies, so you don't have enough of any one protein, and that's certainly the case in plakophilin 2, the idea is that by giving it back, that you're sort of replacing what should be there and isn't. So I don't think that that would be an effective strategy for someone with DSP-related cardiomyopathy who's got enough plakophilin, if that was the question. But if the question was more related to will the plakophilin trials lead to other trials or trials in other genes, I suspect the answer is yes. And I'm sure people will start to look at other areas and other cardiomyopathies as well. And obviously plakophilin 2 is at the moment leading the way because it's the first that's been sort of pushed into clinical trials because of the preclinical research. So I hope that answers the question.
Thank you. How long does it generally take for a treatment to go from phase 1 to becoming available to the public?
It really varies. That whole process that I showed you takes many, many years, maybe 10 to 12 years. But all these things are different. And I think there's increasing interest in speeding the process up while obviously maintaining safety, which is paramount. The study that I showed you right at the end, the N of 1 study, I think took about 18 months from identifying the genetic variation to having a therapy. So these things are definitely getting quicker, but obviously the FDA, their role is to make sure things are safe and make sure that things that we're using to treat patients are doing what we think they're going to do and not going to cause untoward side effects. So that's the real rate limiting step. But again, it all depends on how quickly patients are recruited. Also depends on how quickly it takes to see the endpoints. So if this is something that you can see in a week, the study is going to be much quicker than if it's something that you don't see for 1 or 2 years. So all those things are sort of important in terms of how the study is conducted and designed and conducted, and therefore how quickly it's going to take. But the whole process is a long process.
Okay, thank you very much. So Jeff asks, it seems like most of the studies are with ARVC patients with known genetic markers. Are there studies being developed for those who are gene negative?
Not that I'm aware of at the moment. But with all these studies and everything that happens, all the preclinical research, we're gaining more and more insights all the time. If you told me 5 years ago we'd be looking at PKP2 gene therapy studies, I wouldn't have believed you. So who knows where we're going to be in 5 years' time more. So definitely keep a very sort of open eye on what's happening, because things are changing incredibly quickly. So hopefully we'll be able to expand the different types of therapy we're able to offer in the not too distant future.
Thank you. What are the differences between a drug trial and a gene therapy trial?
So obviously they work in slightly different ways. Drugs we typically think of as something you're going to take every day, whereas a gene therapy trial is a one-off treatment at the moment. And that's partly because of the way the vectors are designed, and the vectors are immunogenic, so they can create an immune response, and modifying that immune response is one of the big areas of research at the moment in gene therapy trials. So you can only have it once. So that's sort of one of the big things at the moment. Obviously, gene therapy is much newer, so a lot of the investigative activity around the trials is very, very intense at the moment, because things are very new, whereas many sort of drug trials have been done for many, many years in different situations. Not so much in inherited heart conditions, because we haven't had different treatments available, so those are coming in. But those are sort of the basic differences that I can imagine, is that this is something you're going to take on a regular basis. It may even be as an outpatient, versus something that's a one and done treatment. But if non-viral vectors become a reality, which I'm sure they will do soon, then there's sort of talks of multiple smaller dosings of gene therapy as one different strategy to adopt. So things are going to evolve over time for sure.
Let's see, many of the clinical trials are done in adults. Is there an age you'd recommend children participate in studies?
That's a great question. And the reason is that most trials have a cutoff at 18 to begin. So entry criteria are you have to be 18 and older. In genetic conditions, you could argue that actually the most severely affected individuals are younger than that, because that's how a lot of genetic conditions work. So why are you treating people with 18 when actually the people who really need the treatment are 3, 4, 5, 6? So what a lot of companies are doing, where they're sort of working with those conditions where there is a sort of much more severe form of the condition younger in life, they're trying to accelerate it very quickly through adults and get it into younger patients as quickly as possible. Obviously, other studies where really it's just children who are affected can go straight into pediatric studies. So some of those conditions are already being trialed in. Some of the immune deficiency syndromes have been sort of looked at for quite some time. So obviously, patients are treated much, much younger in life. But typically for most adult onset conditions, the age of entry is 18, and then it's expanded into pediatric patients from there once it's shown to be effective in adults.
Okay,
but genetics conditions will undoubtedly change that as we move forward.
Okay, Christy comments, how much will the public, or in her case, the disease community, which is patients with ARVC, get to know about the success of trials at various stages? For example, if phase 1 is a success, will it be publicized that the trial is moving to the next phase and has shown positive progress so far, or is that kept confidential?
Well, you could argue that the fact if you see a phase 2 study announced, that's almost by definition saying the phase 1 study was successful and it's been allowed to continue. So that's good news. So I think that many companies now have great patient advocates who work very much with the patient community, who are very much there as a liaison to help you understand what they're doing, the success of trials. So by making a connection with these companies and making connection with the patient advocacy team, that's going to be the best way to find that out, because they're going to be able to sort of feed that back to you directly. Again, you can look at different research studies that may be published in the literature, or often these companies will put that on their website saying, here's our phase 1 data, this is the readout from phase 1, and here it is. So there's lots of different ways that you can find that out, but I imagine the best way is to speak to the patient advocates.
Okay, thank you very much, Dr. Abrams. I don't see any more questions at the moment. Are there any last minute comments to post in the chat field from anyone? Oh, Melanie asked, are certain, so I went to mute here. Melanie asks, are certain people sought out for trials based on their particular situation?
So obviously any trial will have specific entry criteria. So that may be based on genetics or maybe based on specifics of the phenotype that they have. So if you think about hypertrophic cardiomyopathy, some patients have obstruction, some patients don't. So those are important factors. So obviously centers that look after a lot of patients with different inherited conditions will say, when a trial is sort of enrolling, yes, we know we've got certain, 20 patients who may be interested in finding out more about this, and then speak to those specific individuals. That's typically how it works. So yes, so we know obviously what this medication is designed to do, and then speak to patients who may benefit from that specifically. And that's again very important, as we sort of talked about is this trial the right one for me?
Okay, thank you. Deidre asks, what happens if you're part of a trial and you start to experience adverse side effects of the treatment? Can you drop out, or would this depend on the severity?
So I think important, you can drop out of a trial at any time. If obviously, particularly if you're getting side effects from that, but any side effects are immediately reported back to the physicians conducting the trial, back to the sponsor of the trial. So they're looked at very, very quickly and sort of try to understand why a patient has those side effects. Is that related to the agent? So typically, if there's a significant side effect, they might stop the trial at that point and report back to the FDA. Lots of different things will happen. So yeah, no one's going to be made to continue in a trial if they experience side effects that are untoward or unpleasant. So absolutely not.
All right.
And obviously, if one patient's having that experience, then the sponsor's going to want to know about it, because that may be relevant to a lot of other individuals as well.
Exactly. All right. I don't see any more questions in the chat field right now. So I want to thank Dr. Abrams for his time and volunteering tonight to present to us. And I want to thank all of you for your participation and your questions. Thanks for attending. So have a pleasant night. Thank you.
Thanks, Martin. Take care. Bye-bye.
Thank you.